All right, hello everybody. Welcome to Biopsych. We're going to be doing it online. Just for this week's lecture, we're actually going into something that can be very complicated, so it might actually be good that it's recorded so you can go back and review it. And the week after, we will definitely be talking about this and reviewing this again in person. So let me go ahead and get PowerPoint going. Just to review the anatomy of the actual neuron, let's just go over this again really quickly because we're going to start talking about the action potential. So how communication is happening within the cell body. Like before we actually talk about how one neuron communicates with another neuron, we first have to understand how within a single neuron a signal is sent, or what we call propagated. So just to review our neuron, we have the main cell body, which is also called the gray matter. The cell body here, we call it the cell body or the soma. Hopefully you can see my little marker here. Let me just go ahead and go in presenter mode. I can always switch back. The dendrites are kind of the antenna. They're the ones that are listening and connecting to other neurons. Cell body or soma. The nucleus, we talked about this. The nucleus is what contains the DNA. So this is important for writing new proteins, writing new receptors. The axon, of course, is what's going to, where the action potential, we'll talk about that. The action potential is sent down to make a connection with another neuron. An axon collateral doesn't always have to be the case, but sometimes the axon splits off. The axon hillock is where the signal starts, where the action potential starts. And myelin, which is the fatty tissue that the axon is wrapped in, helps insulate, kind of like a wire insulation, make sure nothing else is interfering with it, and it helps with conduction speed. So myelinated neurons, myelinated axons are very fast and very good and efficient at communicating. The spaces in between the myelin is called the nodes of Ranvier. And our axon terminals with the little terminal buttons, we'll talk about this in the next chapter, chapter 2. This is where one neuron synapses on another neuron. So once the action potential goes down, you remember I talked about neural communication being in a electrochemical communication. So electrochemical, the action potential is the electrical part. Once we're at the synapse and neurotransmitters are being sent, that's the chemical part. So chapter 2 is going to be the synapse and the chemical communication. This chapter is going to be the electrical communication, meaning the action potential. So once we're at the synapse, and we'll talk about this in the later chapter, the action potential reaches the terminal button, causes a chemical to be released. This is the neurotransmitter, and that's then what communicates with the next neuron. Okay, so we went through all this. I'm not going to go through that again. Let me go on to what we're going to talk about for this class. This and next, like I said, this is a lot of material talking about the action potential, so I want to make sure we go through it slowly, and because it's recorded, you can go back and view it again, which is great. Okay, communication within a neuron. So here what I'm mostly talking about is the electrical signal that is sent that we call the action potential. So first off, setting up what happens when the axon or the neuron is not communicating. So there's what we call a resting potential, where nothing is happening, no action potential is being sent, but the baseline level of the axon is to actually be negatively charged. So it takes energy to make sure that it keeps that negative charged, but we have a negative charge of negative 70 microvolts. The membrane potential is simply ions. So ions can be positively charged or negatively charged. The configuration of positive or negative ions makes up what type of charge the membrane potential is going to be. And if we have a negative 70 microvolt charge, that means there's a higher negative charge inside of the neuron. So in this case specifically the axon, a negative charge inside of the axon compared to the outside. And we can do this by setting up a recording device, right? Set up electrodes, dipole, measure what's going on here. Okay, so there are two changes that we talk about. Whether the resting potential becomes more positive or if it becomes more negative. So again, resting potential is at -70. If I go more negative than -70, so for example, -80, then we refer to this as hyperpolarization. So it ends up being more negatively charged than compared to the resting potential. And if you looked at what this would look like on a recording device, even something like EEG, you would see here we have time on the x-axis, and on the y-axis we have our membrane potential in microvolts. So here, -70, the dip to -80. That's hyperpolarization. What we're going to mostly be talking about, especially related to the action potential, is depolarization. So something happens where all of a sudden the inside of the axon becomes much more positively charged than before. Resting potential is -70. A change happens where now all of a sudden it becomes depolarized. So more positively charged compared to resting. And what's significant here is that there's what's called a threshold of excitation. So if there is enough of a positive charge that occurs, just from -70 to -50, this -50 microvolts is what we call the threshold of excitation. And once you hit that threshold, it triggers the action potential, which is a whole sequence of events of ion channels opening and closing. So depolarization is when you end up having a more positive charge compared to the negative resting potential. Okay. All right, so what sets up the membrane potential? Why do we have a negatively charged, and how does that even occur? So some of this is going to be some basic chemistry, some basic physics, some basic bio. So again, if you're really not heavy on the science part and you're like, this is not my cup of tea, come talk to me in office hours. I can find more resources for this. But in a nutshell, there are two forces at play that enable this potential, this electrical potential to occur. So the electrical charge of the membrane potential is a result of two different forces, one being diffusion and the second electrostatic pressure. So diffusion, as you can see here, if you drop in a drop of water, for example, salt water, all of the ions and all of the things inside of the water droplet are going to diffuse across the space available to it. So diffusion of moving molecules from regions from high concentration where the water droplet happened to areas of low concentration. The second force at play is electrostatic pressure. So just like I was actually mentioning here, if you drop a drop of salt water into a non-saline solution, saline just means salt water. For example, if I drop NaCl, which is sodium chloride, which is table salt, it's actually made up of positively and negatively charged ions. So Na, sodium, is positively charged, chloride is negatively charged. And don't worry if you're like, I need to write this down. Sodium and chloride, they're both going to come up again because sodium especially is extremely important for the action potential. So electrolytes, this is what you drink in your Gatorade or your Powerade. You basically need more salt in your system because you were sweating and getting rid of salts. So electrolytes dissolved in water will split into its charged particles, cations if it's positively charged like sodium, and anions that are negatively charged. And the force with which they either attract or repel each other, because they're positively or negatively charged, just like magnets, this is what creates electrostatic pressure. And so both of these forces together, as they interact around the membrane, that again, remember, is a bilipid membrane. It's kind of a barrier. This will create different situations. So for example, the bilipid membrane is pretty much will not allow chloride to pass through. You can even see just on this little diagram here, chloride's actually too big to pass through the semi-permeable membrane. Semi-permeable because some things are allowed to pass through, others are not. Some are just too big. Chloride is actually too big, whereas sodium is big enough and it will go slip to the other side. So there's a concentration gradient that's created where diffusion will allow for the sodium to come to the other side. It'll actually remain here because it's being pulled by these chloride ions, so through electrostatic pressure. So this right here showing what, realistic, it's a caricature of the membrane, but both of these forces are at play of how different ions line themselves up around the membrane. Okay. So ion concentrations around the membrane. There's going to be two really important ones that you need to remember. And I'll point those out in a second. So when we say intracellular, this means inside of the cell, inside of the axon. So this right here is going to be inside the cell or inside the axon. We have various things inside. So for example, we actually have organic anions, so negatively charged organic material that's not going to leave. It's kind of trapped inside, as well as potassium, K for potassium. Positively charged ion, it's semi-permeable, so some potassium can go, but as you're going to see, this potassium is actually tightly regulated by the membrane. So these are in greater concentrations inside of the cell, whereas we have higher concentrations outside of the cell of chloride. I was just showing you that in the last slide. Chloride pretty much does not come inside the cell unless there's a transporter or a receptor that brings it inside. And then of course sodium. This is going to be one of the most important guys you need to remember is sodium, Na. Positively charged sodium mostly is going to be outside of the cell. So think of the outside fluid outside of the axon is basically like seawater, right? NaCl, sodium chloride, is basically table salt. So the outside of the axon is kind of a salty environment with the inside being tightly controlled and this membrane separating, and only when it needs to is it going to bring ions in or out of the cell. And for the action potential, sodium and potassium are going to be the two most important ions that we talk about. Just another demonstration of that. So these guys, these are proteins, and these proteins are very specific proteins called ion channels. So they allow, usually it's only for one particular ion. This, for example, is a sodium ion channel. This one over here is a potassium ion channel. So it will only allow sodium or potassium or chloride or whatever ion we're talking about. It's built to only allow the passage of one particular ion. So here you can see sodium coming in, potassium going out. And these ion channels are regulated. So either they open up or they close because of voltage changes, and we'll see that in a few minutes, or they can be opened or closed by something else. And when we talk about drugs, we'll revisit that. Okay, so just again to reiterate, sodium, you can see sodium in red here, right? So this is just our membrane. These green bell pepper looking things are the ion channels. This is showing a closed ion channel. So even if an ion kind of comes in here, it's not going to let it through. This one would be an example of an open ion channel. So sodium in red, much, much more concentrated on the outside of the axon. So sodium 10 times more concentrated in the extracellular fluid. Potassium in gray, potassium is mainly going to be inside of the cell. These two are purposely kept apart. Other things like chloride, pretty much non-existent. Well, I don't want to say non-existent, but mostly are going to be in the extracellular fluid. And then proteins are going to be these little chains because of amino acid chains. They're pretty much going to be absent from the extracellular fluid. Proteins are things that are happening inside of the cell, right? Not floating on the outside. And again, sodium and potassium, these are going to be our two major players for the action potential. Okay, so I said it's vitally important that we make sure in order to have an action potential, we first have to make sure sodium is kept outside of the cell and potassium is kept inside of the cell, right? Concentration of sodium is 10 times higher outside. For potassium, it's 20 times more concentrated inside. We have a very specific channel here. This is an embedded protein, just like an ion channel, but this is a very special one. It's called the sodium-potassium pump. So it allows the passage of two different ions. Specifically, it allows for 3 ions to exit the axon and 2 potassium to come inside. It's always going to have that 3-to-2 exchange. What is so crucial about this guy is that it pretty much takes up 40% of a neuron's energy. So it's active, it needs active transport, meaning ATP. This is the energy that you get from mitochondria. So most of the energy from mitochondria inside of a neuron is responsible, or almost half of it is responsible just for maintaining these sodium-potassium pumps. Make sure we have enough sodium outside and enough potassium inside. So it's going to actively pump sodium into the... Did I write into? I meant to write out. Oh my gosh, that's a typo. I am so sorry. This is supposed to say actively pumps out sodium. Oh yeah, actively pumps out sodium into the extracellular space. No typo. I was just reading it wrong. This takes ATP. This takes energy. And this is what I was mentioning. The membrane is actually somewhat permeable for potassium. It's 100 times more permeable to potassium than it is to sodium. So we call it semi-permeable to potassium, but pretty much impermeable to sodium. Sodium has to be much more tightly controlled than potassium. So sodium-potassium pumps, make sure we are always maintaining sodium outside of the cell and potassium inside. A little demonstration of what that looks like. This side over here is going to be inside of the axon, so the intracellular space, extracellular space over here. Once we get 3 sodium out and 2 potassium, the pink ones, 2 potassium in, 3 sodium out, kicking off ATP. In order for the sodium-potassium pump to work, it requires energy to make this happen. So remember that 3-to-2 ratio. Okay. So I mentioned this, the sodium-potassium pump is a type of protein that is embedded. It's a highly specialized protein. Ion channels are little bell peppers. Like I said, these are also proteins, but they're specialized for the passage of ions, and usually only one ion, like one type of ion. So you have a sodium ion channel or a potassium ion channel. Sodium channels or sodium ion channel can allow really up to 100 million ions per second. So this is an extreme event that happens. When sodium channels open and there's an influx of sodium, it's a huge influx of sodium. And we're going to talk about that, but that's what leads to this huge spike in our action potential. So basically, whether or not you have an action potential is all determined on whether or not ion channels are opened or closed at different points. So open versus closed ion channels change the membrane permeability, because if all of a sudden you have a whole bunch of positively charged ions that come into the cell, then you no longer are at negative 70 microvolts. Now you depolarized and went up to maybe plus 10 or plus 20 or plus 30. So the action potential results from a series of opening and closing of ion channels, and therefore changing the membrane potential. And that's exactly what we're going to go through right now. So this is the action potential. Down here is our resting negative 70 microvolts. If we get some, if the membrane potential gets depolarized, where's my little cursor? If it gets depolarized and it goes from negative 70, here it's showing negative 60. Sometimes different textbooks, different sources have a different threshold of excitation. So sometimes they'll say 60, sometimes they'll say negative 55, negative 50. Any of those answers will be correct. Usually I think I refer to it as negative 55 or negative 50. So a couple slides ago, I said it was negative 50 was the threshold of excitation. So you can just take my word on that. Here, let's just imagine this is negative 50. Once the membrane potential is depolarized enough to where it reaches that threshold of excitation, then it kicks off, I keep losing my cursor, then it kicks off the action potential, huge depolarization. And when you come back down, we actually call this repolarization. It's not hyperpolarization because hyper is only if you go past the resting potential, right? Negative 70 is your resting potential. If you go below that, that's hyperpolarization. But if you're all the way up here at plus 40 and you come back down to resting, we just call that repolarization. So the membrane potential is repolarized. So let's walk through this step by step of what exactly is happening with which ion channel and how are we getting this action potential. Okay, so the first thing that happens, and I mentioned this in here, see I'm like changing the number, negative 55. Once the threshold of excitation is reached through inputs to the cell, we'll talk about that on another chapter, why this even happens. But if the threshold of excitation is reached, that triggers sodium channels to open up. Before that, sodium channels are closed, because remember the sodium-potassium pump is working really hard to make sure sodium stays out. Once the cell registers negative 55, sodium channels open. You can see that here. Bell pepper opens, and then per second, 100 million of these ions are just flying in, right? So sodium enters the cell. Sodium is actually propelled by both forces, by diffusion and electrostatic pressure, into the cell. And this is when we get that huge depolarization. So the membrane potential starts depolarizing. Somewhere around here, during this depolarization, usually around 0 microvolts, is when potassium channels now are also going to open. They were closed beforehand, but this depolarization leads to potassium channels now opening up. So in our little blue bell peppers, this is a potassium channel. Potassium channel is open. And because potassium was being kept inside of the cell, it starts flying out of the cell, right? So sodium in, potassium out, the opposite of the sodium-potassium pump. Potassium flows out of the axon membrane. It's also being pushed by diffusion and electrostatic pressure. And it basically, this entire process of sodium in, potassium out, continues until we reach plus 40. This is basically the max membrane potential for an action potential. So once plus 40 is reached, something very significant happens. Those sodium channels all of a sudden close and stay closed until we reach resting membrane potential again. So they will not open no matter what until the voltage comes back down. So plus 40, sodium channels become refractory. Refractory just means locked in place, closed and locked. Channels become blocked and cannot open again until resting potential is reestablished. So sodium no longer comes in, but in the meantime, what's been happening is that potassium is still leaving the cell. Potassium channels are very slow to open and to close. So they're just kind of lazily like, oh, sodium's doing its thing. Let me open up too. And so during that process, sodium tightly controlled, threshold of excitation reached, opens up, Sodium comes in, plus 40, sodium channels close. Very like punctual and very, very fast responding. Potassium is like, oh, I guess we're opening up. Slowly start doing that. And during this process here, when my little red arrow started coming back down here, it's actually repolarizing because potassium is still being driven out of the cell. So that overflow of potassium still leaking out is actually going to cause the inside of the membrane to become negative again. It's going to continue doing that until we come all the way back down to resting potential. So here you can see potassium leaves the cell, causes the membrane potential to return to its resting level. Very slowly, again, during this whole process of repolarization, potassium channels start to close. So they were late to the game opening, they're late to the game closing as well. Once we reach resting potential, then theoretically, well, not theoretically, once we hit -70, sodium channels reset. So they were refractory, they were locked in place. Once it hits -70, they could open. It's not to say that they are going to open, because it would need an input for them to open again and reach threshold of excitation. But it resets, they are no longer refractory. So the refractory period is actually the period from here, when they become refractory, plus 40, to exactly -70. That's called the refractory period. No new action potential can be triggered during that period. Okay, so the resetting allows for a new action potential to come through. If that's what's happening to the neuron, if like a whole bunch of other neurons are talking to it and saying like, let's have a party, then at this point, it could send another action potential. Again, potassium channels are slow to reset. They're slow to close. You can see that here. Theoretically, a sodium channel could open again. A potassium channel is like, I'm sort of closing. What happens is, because it's so slow, we actually end up having an overshoot. So more potassium's leaking out than it should, and this actually leads to a hyperpolarization. So that's what this bump is below the resting potential. And then the last step is that extra potassium kind of diffuses away, and then the whole thing gets reset again, right? During this whole period and beyond, a new action potential can occur. So sodium potassium pump helps then resituate all those ions. And I'll talk about that in a little bit more detail in a second. So that is the action potential. Like I said, it's probably actually good that today's lecture is recorded so that you can go back and view this. But let me show you a few more graphs for you to really kind of solidify what's going on here. So here's, sodium is in blue. This is outside of the cell. Threshold of excitation is reached. They fly in here, while at the same time, pink, which is potassium, leaves the cell. That's the potassium that slowly kind of exits the cell, right? And you can see over here what happens when sodium comes in, depolarization. When potassium does its exit thing, it leads to repolarization as well as a hyperpolarization, right? So keep in mind, while sodium is coming in, actually around here, potassium is already leaving. But the main up and down of the action potential is being driven by sodium coming in and potassium coming out. That's why it's shown here so dramatically as two separate events. But keep in mind that the opening and closing of potassium channels is slow and ongoing before we actually reach the peak, meaning plus 40. You can see that the opening of sodium channels, much more fast. Nothing happens until we are reset, right? And once we reset, then they could theoretically open again. Okay, one thing to mention, so I said what you can see here, right? We start with blue on the top and pink down here, and then they change their positions. So pink leaves, which is potassium, pink leaves the cell. And now that the action potential is over, it switches, right? Blue's on top again, pink's on the bottom. It's the sodium-potassium pump that's responsible for reconfiguring those ions, right? All of that sodium that came inside, make sure we pump that back out. Use energy and pump that sodium back out. But one thing to keep in mind, just perspective-wise, the action potential only causes 0.0003% of an increase in sodium inside the cell. So even though I said is 100 million ions per second, also keep in mind, I don't think I put on here how fast this is occurring. Do I not have a, sorry, here it is. So just so you have a time scale, the action potential takes less than 2 milliseconds, right? So if it were open for an entire second, then yes, you would have more than 0.0003% of sodium coming in. But this is a fraction of a second. Already after 1 millisecond, sodium channels have become refractory. By, what is this, like 1.5 milliseconds, another action potential can be sent. Okay. So just to put that, make sure that we were talking about the time scale here. So small, very fast, small event that happens, only small percentage of sodium comes in, but so this sodium-potassium pump is more of a long-term functioning. Make sure that everywhere throughout the brain, all neurons have that nice sodium outside, potassium in setup. So it's more for long-term, not for short-term functioning. And the refractory period is important for when we talk about where the action potential actually travels, right? Because it's electricity. Electricity, just like lightning in Tampa, is going to go in any direction that it can. So an axon is just one long thing. If an action potential happened in the middle of the axon, which it doesn't, but let's just say it did, if you like triggered an action potential, it could go both ways. But what's happening is the action potential is triggered at the axon hillock, and it travels down. The reason it only goes in one direction is because of the refractory period. Because at some point, from that plus 40 to that negative 70, no sodium channel is going to open up here, right, where the action potential already occurred. So I'll show a few other pictures so that you can kind of wrap your head around that. So when we talk about the action potential, it travels all the way down the axon. So it's an event that happens on one location, but then it travels down because as it's moving, it triggers other sodium channels to open and other potassium channels to open, and it travels down. So you can see if you stuck recording electrodes along the axon, you would see, at the beginning, you're going to have your action potential. At the next point, it'll be a little bit later. And the next, so it's a traveling electrical event. So we say it propagates. It propagates along the axon. So it retains its intensity. It is an all or nothing event, right? If threshold of excitation is reached, it doesn't matter what else is happening. It will trigger those sodium channels to open, and there's no stopping it. That sodium influx causes that depolarization, and it triggers potassium channels to open. So once triggers, propagates down the axon, there's no stopping it. And you can see here the axon hillock. This is where it starts. Okay, here's where I can show that a little bit better about the refractory period, why it only goes in one direction. This is from your textbook. You'll notice a lot of the figures are not in the textbook. These are from other textbooks that I've used in the past to try to tell the story a little bit, because it gets complicated when we talk about the action potential. Okay, so here, stimulus will start up here. Let's say this is the axon hillock, where the action potential starts. Sodium here exiting, potassium coming in. And then it basically continues down. So we have other ion channels here that are sitting. If this is occurring here, this electrical event will continue to the next person. So it's going to go trigger these guys to open, and then less than a millisecond later, it triggers these guys to open. But the reason why it can't go backwards is because if here the sodium channels are open, and theoretically, I'm like trying to touch the screen and you can't see me touch the screen. the signal could go in both directions, not just go down. But if it were to come back up here, these guys are refractory. These sodium channels are not going to open because they're in the refractory period. So in the only direction that the electricity can have any effect is this way, where sodium channels are not refractory. And they are waiting for the threshold of excitation. And when that potential reaches it, it triggers them to open. So that's why it only goes in one direction, because of the refractory period. Okay. Maybe I'm talking faster than I should. It's already been, I think, 30 minutes. Okay. So I mentioned that an action potential is complete after only 1.5 milliseconds, right? And if there is another input, it could trigger a new action potential. So now the question is, if the only way neurons communicate electrically is through their action potentials, how do we code for, blue, purple, polka dotted t-shirts? Like, how do neurons do that if all they do is just send action potentials? So we refer to this as coding of stimuli. So we have neurons that only do visual coding, neurons that only do auditory coding, or motor coding, right? But when we want to have a neuron tell you what type of stimulus is this, you have to do it through the changes to the action potential. So the variation in stimuli, so let's take, for example, the visual system. We have a dim light versus a bright light, or you have to move your muscles a little bit to pick up a pencil, or you have to move your muscles a lot for weightlifting, right? So there's a difference in the strength of the stimuli that we're talking about. Someone's listening to music quietly, or they're blaring it in their ears. And so this is coded not by the size of the action potential, but by how quickly you're going to fire an action potential. So we call this the firing rate, or the rate of firing of action potentials. So for example, here, each line here represents an action potential, that depolarize and repolarizing event, right? The thing that happens like in a millisecond, that's represented here by one little blue line. So let's say you turn the light on first thing in the morning, and maybe you have a dimmer, and you turn it on as just like a gentle little night light or a night light or something like that. Your visual neurons are going to code that as action potential, just like a little bit, right? Coding maybe, let's say, every 10 milliseconds. It's going to send another action potential because you're still getting that input into the eyeball. So it's going to say every 10 milliseconds or so, right? If, however, your roommate comes into your room and you're not ready to wake up yet, and they turn the light on and it's super bright and obnoxious, your visual neurons are going to code this strong stimulus with much more rapid firing. So not every 10 milliseconds, but maybe every 1 or 2 milliseconds. So this is called the rate of firing between a weak stimulus and a strong stimulus, right? Or if you have to lift a pencil, muscles are going to code this through sporadic or intermittent or greater intervals between the action potential. Whereas if you have to pick up, if you're doing a bench pressing for like 120 pounds or something, your muscles are going to be sending much more frequent action potentials. Okay, so this is called the rate law. This is how things are coded. The action potential are -70 to +40, back down to -70, hyperpolarization a little bit, and then resting potential. That shape never changes. So if you have a heavy weight that you're lifting, the action potential doesn't just go all of a sudden up to +100, it will still only go to +40, but it'll do that more frequently, right? So this is how we code for different things. Because there's such a delicate balance in ions going in and out, we don't change it for the different stimuli. We do that by how the action potentials are actually fired, how frequently they are fired. Okay. Okay, signal propagation. So we mentioned this when we talked about the anatomy of a neuron. You have your myelin sheath, right? But it's actually wrapped in little chunks. It's not homogeneous along the entire axon. It's wrapped in little chunks. And the reason for this is the little space that's left in between, and here it's blown up. Here's the myelin sheath. This is the raw axon underneath. The space in between, that's called the nodes of Ranvier. Allowing for space like this, think of it like if you like racing games and you go over those little like speed sections on the floor where if you drive over that, you're going to get a little speed boost. That's exactly what the nodes of Ranvier are. So in the myelinated regions, in here, no exchange of ions can happen. So no sodium's coming in and no potassium's leaking. It's entirely insulated, which is good so that it doesn't disrupt the action potential. But if you wanted to send it down, like let's say a 2 meter long, or no, but 2 meters is a little big. Let's just say like I'm really bad at estimating things. A really long axon. If you want to send the signal down a really long axon and you insulated the entire thing, it would be slower than if you broke it up and allowed it to get a little speed boost at the node of Ranvier. And the way it's getting a speed boost is because it's not myelinated. This is where you can actually have sodium-potassium exchange happen because it's exposed to the extracellular space. Here, nothing can happen. No new sodium can enter, but you can get a little booster here where new sodium can come in and potassium out, and it boosts the signal, and it kind of like almost jumps down the axon. So nodes of Ranvier are the boosters. We call this saltatory conduction because it's saltatory because of the saline solution it's sitting in, right? I said sodium chloride is on the outside. So you actually kind of, because of salt, saline conduction, that can help boost the signal down. And on top of it, it doesn't just help the speed. Remember those sodium-potassium pumps that are energy-wise very expensive for the cell? You don't have to put them in here. Sodium-potassium pumps do not need to be in here because they don't need to reestablish any kind of balance. So you can save sodium-potassium pumps by only putting them at the nodes of Ranvier. So it's more economic. You don't have to have the neuron working overtime. Just where ion exchange is happening is where you put your sodium-potassium pumps. So, highly effective system doing it this way. Okay, so that was very fast, but yes. Wrap up things to think about. So, what forces are at play when we are doing this membrane potential establishment, right? Think of one of those first few slides. So we're talking about electrostatic pressure and diffusion. This is what's going to make, just because a channel opens doesn't mean the ion has to go through it. But because there are chemical gradients and electrostatic pressure and all of these forces at play, when the sodium channel opens, those forces are actually going to enable it to just fly into it. And same with potassium, but potassium is a little slower. How do sodium-potassium pumps work? Know that 3-to-2 ratio. It uses energy. It's very costly. Where would you find sodium-potassium pumps? Your major ions that are at play. Sodium, potassium, of course. We do have some other ions that are at play. Not necessarily that are at play for the action potential. So definitely know that configuration, that step process of when, from the moment that the threshold of excitation is reached, which sodium or which ion channel opens, sodium channels open, what is the change in voltage at each point, and when are different ion channels open or closed. I want you to be able to write down that entire process, right? In kind of like a 1, 2, 3, 4 step. I think it's six steps process. And yeah, describe the exchange of the ions across the membrane, different points. Yeah. So I think I have a video, a YouTube video where they show like a graphics way of how these ions are moving and how it's affecting it. So I'll see if I can dig that up, and then I'll attach that into Blackboard so that you can view that. But definitely practice this, without looking at the book. Can you write these steps down? Because there's a lot going on here. Okay. Cool. So that was it for communication within the neuron. So basically the electrical event, the action potential, Chapter 2 then is going to be the synapse. So when we talk about neurotransmitters. So this whole chapter was talking about what does the neuron look like, how is it configured, what are the different parts of it. So there's going to be in the in your brain coloring books, there are a few pages where you can look at the neurons. So I will put a homework assignment up on Blackboard for you to do that. Basically, color in the neuron with the different names that we learned for the different parts, right? These guys here. And then you'll basically just take a picture and upload that. But you can find the information on Blackboard, and I will send an announcement just clarifying specifically what I mean here. So hopefully this was informative. Hopefully you can go back and review this. If you find this actually useful, I can do this for other lectures as well, even if they're going to be in person, right? Obviously everything's going to be in person. This was an exception where I couldn't do it in person, but sometimes I find even just audio recording can be helpful for students. So let me know what you think. I know obviously we're all happy to not be in the pandemic days anymore where we don't get to be in person in class, but the extra information can be useful because you can rewind it. Okay, all right, so hopefully that was good. That was 45 minutes, so that might have been a little fast, but you can go back and review it. So I will see you guys next week in person. All right, thanks guys.